negative comments. Well, indifferent comments made about insects. I used to be an insect indifferent as well until I started on this project, and I thought I've got to learn something about insects. And so I got this book, and I tell you, I am a convert. They are the most amazing animals, and if you love nature, you've got to find out more about insects. They're quite amazing. So, and anyway, I'll show you on the talk. I hope you're convinced afterwards that they're great. Okay, cheers. Okay, so there's three groups involved in making this project happen. There's the SSEC, where the volunteers who go out and do the collecting every week at 4 sites in the national park of the insects. And we see this as supporting the NPWS Eco-Health Scorecard approach. You can't protect what you don't know exists. And so we're adding a lot in terms of knowledge about insects to the inventory of the mammals and the other vertebrates. But the heavy lifting of the project is really done in the University of Guelph within the context of the International Barcode of Life. So that's the lead-in.
Just swish. So,
okay, I'll just mention first up the people involved in the project. So we've got Bria McClafferty, who is our Eco-Health Scorecard liaison person, and also a co-collector, and I would say now a co-enthusiast for the project itself. And then we've got the Sutherland Shire Environment Centre volunteers, Melissa Harvey, who is here off the back and knows a lot more about genetics than I do. And we've got Sue McTaggart and myself from the SSEC. And then my colleague, Paul Hebert, who is really the brains behind this international project with his colleague, Kate Perez. And then Anthony Chariton and Jana Shaw from Macquarie University. They help us set up, geolocate the sites, and also with some of the provision of 95% alcohol, which is extremely important for the preservation of the DNA in the samples that we collect.
Ross, can we just check that the people on Zoom are getting, are you onto the second screen on Zoom, which starts with the people involved?
Yep.
Yeah, they are. Okay, thanks.
So I'll introduce Paul Hebert. So this is the personal connection that I had with Paul. I was a clueless honours student in zoology back in 1973, and Paul had just done a sterling PhD at Cambridge University. So he came back as a whiz kid. He's been a whiz kid ever since, but he's a really nice guy. He's a Canadian. And we used to go down to Bowler Creek looking for powerful owls in 1973. So that's how I got to know him. And then I left, I got a job at ANSTO, and he went somewhere else and ended up back in Canada, running all sorts of highfalutin institutes, etc. So Paul is in his late 70s, but he is committed to this project of the discovery and identification of all multicellular life on Earth using DNA to do it. And he's got the money to do it. He's got $180 million from the Canadian government to do this worldwide. And he keeps getting more philanthropic money as well. I think he just got another $20 million. So he's quite a character. He works 7 days a week. He said, Ross, my own biology will retire me. And so he's never going to retire until he dies. So, you know, it's sort of higher purpose stuff with him. And it's so inspiring to work with people like Paul. It's just amazing. So, and he's, as I said, a Canadian, a very nice person to work with as well. Now, I just want to mention something about insect biodiversity. It's a real concern within the scientific community at the moment because of this insect crisis. This massive decline in the insect biodiversity around the world where it's been properly measured. At the same time, we're finding out about all the ecosystem services that are provided by insects. Or, you know, nearly all the pollination, the pest suppression, the decomposition of carcasses and things, it's all done by insects. So you can go nicely walking through the national park and you're not tripping over a carcass of something that's slowly decaying due to bacteria, the domestic beetles, etc., are getting stuck into it and rapidly removing it. So, most importantly, the stable food sources for other species like birds and mammals that we tend to care more about.
and
So,
platypus.
and platypus as well. So it wouldn't happen, these ecosystems wouldn't happen unless we had the biodiversity and the biomass. So they're both important and they're both in decline. So here's just a few what I call biodiversity realities. Most biodiversity on Earth is terrestrial. It's about 10 times higher than what you get in the marine environment. 70 to 90% of terrestrial biodiversity is insects. And most of that insect biodiversity has wings, okay? And so what we're collecting with our sampling technique, it doesn't get them all, but it gets most of the terrestrial biodiversity. To come back to some of the science that underpins this concern about the decline of insects. This was a paper that came out, I think, in 2017, and it was based on data that had been collected at 63 protected sites in Germany. So not agricultural land, not disturbed habitats, protected habitats like national parks. And the Germans, being very systematic and very thorough, had been monitoring with these Malaise nets, which are exactly the same ones we're putting in the Royal and sampling. They found a 75% decline over 27 years in total flying insect biomass in the protected areas. So this is solid scientific data, and it's really quite frightening. And it's not only happening in Germany, it's happening in other parts of the world as well.
Have we got a baseline to measure? If that's of 27 years, they've obviously had a baseline of 27 years.
They had a baseline.
Have we got anything?
I don't believe we have a baseline, but I mean, anecdotally, if you've ever driven up the coast with your parents when you were young, the windscreens were covered in insects. You drive up now, you get one or two. So that's a sort of an anecdotal comparison, but
Even bush flies now are greatly reduced. Some are, and we think it's great.
I think it's great, but it's in terms of ecosystem functions, it's terrifying.
Yeah,
and pollination, etc. So anyway, so that's a very pivotal paper. So if you look at some of the data that was measured here, and note it's on logarithmic or geometric scales, you can see the decline through time. If you look at it, the annual sampling programs, you'll see there's a hill here for the Northern Hemisphere, and see the colours change as you go down in biomass. In fact, what we will have a valley from our sampling, because we're doing it every week at 4 sites. So we will have something like that as a baseline, a contemporary baseline. And when it's all put together, you can see this quite dramatic decline.
Can you explain what you mean by the hill and the biomass, just for the more dense people in your audience?
Well, this is German data, so you're in the Northern Hemisphere.
Yes.
So you expect to get the highest biomasses in the summertime,
Yes.
and then it drops off in the wintertime.
Okay.
And so this is
colours meaning
the different colours. Well, this represents 2017, and
okay.
that's 2017. So you can see this decline, this trend of decline. Right. Now, just to bring it back to self-interest, if you're still an insect indifferent, everybody likes chocolate, loves chocolate. If you love chocolate, you've got to love the chocolate midge. It's about 1 millimetre in length. Things we don't see, but they're doing all these great jobs for us. So it's absolutely important in terms of pollination of the flowers for the cacao pod, making the chocolate, and of course, out of that comes these really nice films, Chocolat, with Juliette Binoche and Johnny Depp, Judi Dench. So that brings us back to the self-interest issue of the importance of insects. So you can love insects. I just want to mention my first job at ANSTO was my first research project at ANSTO. Ended up, I have a bit of a love-hate relationship with ANSTO. My first job was to dissect 737 fish, purple striped gudgeon. And so for a whole year, I was sitting down every day with a dissecting microscope, identifying the gut contents, and then spent a couple of years doing all the statistical analysis, and it finally came out in that paper. But that's a long time to be looking down a dissecting microscope. So
are they a native fish, Ross?
Yeah, they're a Northern Territory fish. And there's other ones around here, but they're related, but they're similar in size and taxonomy. Anyway, it's just a personal note. Okay.
So that was talking about mining pollution?
Yeah,
last week?
Yeah, that was mining pollution. I was just talking about, I have a love-hate relationship with midges, that's all. That's got nothing to do with what I'm talking about in general. It's just one of these digressions. Okay, let's get back to something that is significant. And that is a really good data set put together by Cornell University to show bird population declines in the US by 29% since 1970, and especially for the insectivores. And so it's a dramatic decline and probably related to the decline in insect biomass and biodiversity as well. If we look at that data dissected into different habitats, we can see it's up over 40% for coastal regions, but there's declines in many sorts of habitats over this period. And so it is a really terrifying result. You know, if you love birds, then you should be concerned. But that sort of brings it into the focus for the vertebrate lovers rather than the insect lovers. Okay, so now we want to get onto the barcode issue. What is a DNA barcode? How do you get it? It's a method of specimen identification using a very short area of the DNA. And so this schematic shows you take an insect, you take a leg off, you do the DNA, and from that you construct the barcode. And then they're put into a reference library. And so we've got about 10 to 20 million species, and the aim is to get a barcode for every one of those species. Okay, this is the logistics of getting the barcode. It's about $3 to construct the barcode, and the technology is getting cheaper and cheaper all the time. And so this is a great focus, a big focus of Paul Hebert and his team to try and reduce the cost of the barcode development. And so after we've collected the samples systematically, we send them over to Paul and his team of 150 people. Believe it, 150 people has got working on this. It's incredible. They put the individual specimen into the database, curate the specimen, photograph it, take a tissue sample, extract the DNA, do PCR, polymerase chain reaction to develop the barcode region, sequence it, and then validate the data, and then they put it into the Barcode of Life database. So as I said, he wants to do all multicellular life on Earth, and he's got the budget to do it.
Sorry, Ross. So all of that costs $3.
Yeah. That's
Seriously?
That's the cost recovery of that, yeah.
Per species.
Per individual.
Yeah.
So when we go out and collect, for a week from one sample site, we'll probably get 500 to 700 individuals. That's why he's got 150 people working for it.
Seems incredibly cheap.
It is amazing,
yeah.
So he works on the mitochondrial DNA because it's very conservative. And so as opposed to the nuclear DNA, which is recombining and crossing over and whatever, due to sexual reproduction, the DNA in the mitochondrium is quite conservative within a species, but it varies between species. So it's really good to identify each individual species. And a certain component of the mitochondrial DNA called cytochrome C oxidase is what is focused on in terms of creating the barcode. Melissa, correct me if this is wrong.
I'm a bacteria person. So.
Oh, okay. Yeah, so that's the details of it. And so this is what a DNA barcode actually looks like for this species of wasp, a North American wasp. And that's what goes into the database. And as I said, there's going to be 10 or 20 million of these barcodes. Now, what I should say is that taxonomy is changing all the time, whereas the barcode is forever. Once you've determined the barcode, you've got it in the library, it's not going to change. Whereas there's not enough taxonomists around to get the job done with respect to the rates of decline in species. And so this is a way of quickly getting a handle on what biodiversity we have, put it in an archive, and then you really know what you're doing with respect to biodiversity conservation or loss. So why is this being done? It's because of what's called the Linnaean shortfall. We've only identified with Linnaean binomial names about 25% of all species on Earth. So talk about biodiversity, we don't really know what we're talking about. So this is to correct that lack of knowledge The other issue we don't know much about is the so-called Wallacean shortfall after Alfred Russel Wallace. We don't know enough about the geographical distributions of all these species as well. And so, you know, Paul Hebert and his team are going to cover the whole world. They are covering the whole world. And we're part of that, and we're the East Australian temperate region. Okay, so just say something about the use of the DNA barcodes. Our barcode reference library is very incomplete. And so this impedes the application of inexpensive DNA-based screening methods like environmental DNA, metabarcoding. Yeah, that was used in the upper hacking to look for platypuses and for their diet, etc. So this is a way of helping that process. And so the project is certainly addressing this gap by barcoding our collections that we've been sending over for the last 6 months. Okay, so this is the sort of trap we're using. It's called the Malaise trap after Rene Malaise, a Swedish entomologist from the early 1900s. And he created the net and spent a lot of time in Yangon in Burma, catching hundreds of thousands of insects. And so it's a technique or a net that's used in many parts of the world. And they were setting up, you might recognise some of the individuals there. We're setting up one of our nets at Little Marley, for example. Okay, so at the moment we've got 4 nets. They've been in place since late December, early January this year. One's at Little Marley and Marley in the heath areas. And I must say they require constant vigilance and occasional repair during the strong winds, which can blow them over. So, but over time we've got better at placing them and stopping them being blown over so frequently. So that's what they look like.
So how do they work?
Well, in the middle of this is a curtain, a black curtain. And so the insect tend to fly in there and then they move up to the lighter areas and they get funneled into this plastic container. And then they finally get exhausted and fall down into 95% alcohol. Yeah.
So the sides are open too?
The sides are completely open, yeah. And we've got one at Lady Carrington Drive and another one at the Ranger station. So we go out and we sample them weekly and prepare the samples and then ultimately send them over to the University of Guelph for analysis. This is the sort of information that comes back to us. Every individual that we send is photographed and then a piece of its leg or something's taken off it, and then they do the DNA and then they do the barcoding. And so it's very labour intensive for the taxonomist. So apparently, Paul, who's got 150 people, he's got one half allocated to the taxonomists and the other half allocated to the DNA people. And so I'm sure they talk to each other, but that's how it's organised logistically in this lab. So there's 150 of them working away on characterising the DNA barcodes of all multicellular life on Earth. Quite remarkable.
So you only send, when you send a sample to him,
yeah,
it has to be something that you know what it is.
No, we don't really have to know
Right.
what it is. And
Okay.
I must say that I didn't know much about insects or even really respected insects so much, but now I do. After working with Paul and reading more about it, and etc., now I realise the significance of this project because it supports everything else. All the birds, all the mammals, the reptiles. You know, ultimately it comes down to these insects and their abundances and diversity.
So you could be sending him something that nobody's ever seen before.
Well, I'm coming to that. I'm coming to that.
Do they test every single individual that you send them, or do they go through those individuals and find, this is all the same one, we test one of those?
And my advice is they do every one. Yeah, okay. And this is an example. This is one page of 28 pages, I think, of the results. This is a phylogenetic tree based on the genetic similarities and differences. And so they're categorized according to their taxonomic position. Just to look at that in easier detail, the Hemiptera are the bugs, Hymenoptera, they could be the bees or the wasps, and then we've got the spiders, other sorts of arachnids, Neuroptera, lacewings, and a few beetles as well. So that's the sort of detail you get coming back for 7,000 specimens that they'd characterised. And so it's all in a big family tree as well.
To Paul's point, they do every single thing that you send over. So
Yeah.
for one of those, there could be 1,000
Sure.
examples of that. Yes.
Yeah, sure. Yeah,
Gotcha.
that's the intensity of the examination of the insects that we're sending.
They're sorting
Is that
that.
a-
You're not sorting.
Is
We're
that
not
a
sorting
family-
that.
Is that a family, genus, species?
Well, yeah, there could be orders or families. Yeah, orders or families.
So for each family, there would be literally possibly, over 1,000
Sure.
down the tree.
Yes, exactly. Yeah, but when I say that, it's a coleopteran, but it may well not have been identified taxonomically at this point in time. But it has a unique DNA barcode. And as I said, the upper level taxonomy, a lot of that changes all the time according to how the taxonomists feel on the day, sort of thing. But the DNA barcode is permanent. It's a constant. So this is from our first 5 weeks of sampling. Okay, trap 1 and trap 2, Marley and Little Marley. And from that, there were 7,000 specimens odd. And the bins are the barcode ID numbers, in other words, the species identified according to the DNA barcode. Marley and Little Marley, they're only about 5 kilometres apart, okay? They're both in Heath. But you can see that from this Venn diagram that there's a lot of independence in terms of the biodiversity of the insects. There's a bit of overlap, but they're quite independent. So, and as Paul said, many species are likely undescribed, which means they're probably species that have never been found before, identified by taxonomists.
So of your 7,000 species that you sent
Individuals,
over,
yeah,
you've got 900
and
different
91
species.
species, yeah.
Oh, there's lots of duplicates, presumably.
Yes.
Yeah, I suppose so, yeah. So, maybe 1 in every 8 is a different
Yeah.
species, yeah. If you plot the number of individuals against the species diversity, these plots, okay, they're not asymptotic yet. So there's a lot more still to be found, yeah. And I just want to tell you about the results that arrived last week from a lot of other samples we've collected and sent. And so we've got our 4 sites here, Marley, Little Marley, Ranger Station and Lady Carrington. The 2024 samples that we collected in November, December, they've all been analysed. And 2025, about half of the samples that we sent have been analysed so far. And you can see the ballooning of the numbers of new species that we've found. And that's only for about 1/3 of the year. Yes, quite remarkable.
So that's new species from their DNA identification.
From their DNA barcodes.
Yeah, but not necessarily, they might be known to entomologists.
They mightn't be known to a taxonomist, but they're certainly known to a genomics person now, because that barcode doesn't change. The upper level, the names that are put on things, they may change because that's what taxonomists do, but the DNA is forever. The barcode is forever.
Did you do less sampling in 2024 than 25?
Yeah.
Why are the circles?
Well, we started sampling in late November, early December.
Okay, so it's a
Yeah,
smaller
so they were the
sample.
first ones we sent over.
And it looks like you only
Subsequent to that-
sampled at 2 sites.
That's
24 as
That's
opposed
true,
to-
that's
yeah.
true. And then we got the other ones in place in about February,
Yeah.
and we've been sampling weekly since then at 4 sites.
Yeah, okay.
So we've got 3,839 species for about 1/3 of the samples. And so if you project, and Paul is quite tentative about this, he doesn't like to exaggerate things, but based on the current data assessment, there's probably about 10,000 arthropod species. So there's insects, but there's other things like springtails that are not really classified as insects. They're something slightly different. And, the spiders, etc. So there's probably 10,000 arthropod species just by that one sampling methodology. So if I push him, I say, what does this mean, compared to other parts of the world? He said it's at the upper end of biodiversity for temperate regions around the world. So that's our Royal National Park. It's quite biodiverse in terms of and so, there's lots of other interesting things to find out. Yeah, that's it.
Is there a less destructive way, or what's the impact of the sampling method on the populations? Is it a drop in the ocean?
I think it really is a drop in the ocean.
Okay.
Yeah, I really do think it's just a drop.
Like
I mean, those sorts of things concern me,
excess.
but there's so much out there. I really don't, and their reproductive capacities are so great that I don't think what we sample is more than maybe 1% or something of what is out there. So,
Unless it's a rare one-off.
oh well, I suppose that's always possible. You can always posit that, I suppose. Yeah.
Sorry, another question.
Yeah.
What's the difference between a barcode and the genome of the species? Is it the same thing?
Well, I mean, if you're talking about the whole genome, then we're only taking this cytochrome C region of the mitochondrion because it's quite conservative from the mitochondria. So there's a lot of the genome that isn't being looked at. It's only this region.
But it's unique enough.
It's unique enough because it's conservative within a species, but variable between species.
So why don't they just look at that instead of whole genomes?
Well, it depends what you're looking at it
Yeah,
for.
that's true.
If there's a whole lot of reasons to look at different
Absolutely,
components.
yeah.
Yeah. And as I said, I've got limited genetic knowledge, but ask Melissa, she's closer to this stuff. So now I just want to visualize some of the things we've caught, they're microscopic and you sort of, bloody insects, who cares? But when you look at them close up, they're really quite beautiful. So the coffin fly, for example, it'll dig down 6 foot looking for a cadaver, incredible. Houseflies, there's all sorts of species of houseflies. They're quite biodiverse. Midges we know about, they're bloodsuckers. Crane flies look really nice. They don't do many terrible things to anybody, I don't think. And the jackal fly, it lives its life by jumping on the back of a spider and sucking on its body fluids.
Lovely.
So yeah, so that's the way to earn a living. So I'm talking about the things we catch most of. So we catch a lot of the flies and we catch a lot of the wasps, bees and insects. So even though it's not a flying insect, we still capture it. We still get it in our nets. These little guys, these fairy wasps, they're only 0.5 millimeter in length. And for them, air is like paddling through honey. And these little wings are like paddles. They use them as paddles. And they flick them 1,000 times a second. It's quite remarkable. This is what I mean. I'm getting excited about insects and I never had liked them before. This braconid wasp, it's a cute little thing, isn't it? With the big eyes. Yeah, lovely. But if you're a caterpillar, you don't really like it that much because it'll kill you. And these guys, these fairy wasps, they're very important in biological control as well. So these other things don't increase exponentially in numbers. And the Darwin wasp, Darwin loved insects, So the Darwin wasp, there's 24 or 25,000 known species of Darwin wasps, ichneumonid. That's as many as all the mammals and birds put together. So it's a lot of stuff happening. Well, they've been around for, since the Devonian, so they've been evolving and speciating since then.
What's killing huntsman spies and dragging in the water?
And these
Those
guys,
ground
when they
wasps.
raise an egg,
Amazing.
yeah, they're incredible. Yeah. I just put the moths in. We catch quite a few moths as well. So they look pretty nondescript, but every one of those moths is from a different family. So there is different genetically from each other as a goanna and a blue-tongued lizard would be. They're different families of reptiles. So yeah, we catch quite a few of those. They are probably more important for pollination than butterflies are. The good old moths that we typically don't think much of. So now I've got some better pictures that have recently come back from Paul Hebert and his mob over there that I just wanted to share with you, the aesthetics of insects. Here's a nice fly. I call it a charismatic fly. It's iridescent. It's got lovely, great big compound eyes. When you see it like that, I think it's a thing of beauty. I see people nodding their head. That's great. Here's a weevil. Again, you see them up like this. They're sort of quite remarkable. Beautiful looking things. And this is a lacewing. you look at the size of this, it's only a few millimeters in length. It's a beautiful photo. You can see the sort of the beautiful anatomical design of these things. Remarkable, yeah. And here's Icumonid. So these are all individuals that we have sent to Paul. So they're our insects in the park. I just wanted to share some of that nice photography with you. Okay. So also along the way, we get a lot of bloodsuckers. A lot of the females will feed on blood. They need the protein to make the young. So horseflies, midges, and also mosquitoes. Anthony Chariton and his team have been doing the mosquitoes with NPWS alongside their scorecard collection sites. And we've got our nets near the scorecard sites as well. And so they've done the blood and they're looking for mammals and birds, etc. In the blood. But these other guys are feeding on blood as well. And Paul and his team are now working through some of these samples. They're going to be collected over a much longer period to see if they can detect any vertebrates. So we could find something that's never been found before just by looking at the blood of these bloodsuckers. So, okay, so now I pose a question. Should national parks be even further valued as repositories of insect biodiversity that restores ecosystem services to biodiversity degraded agricultural landscapes adjacent and beyond? I say this because of their capacity for great increase and their abilities to migrate so much. So, that's another component. Now we've found all this great biodiversity of all these things that can make a difference. It's another string to our bow with saying national parks are so much more important than we thought they were before because of all this species biodiversity of insects living in. This is the Royal National Park. It's the first time it's been done.
Yes, is the answer.
Oh,
Yes.
good. I'm preaching to the invertebrate, of course. Now, I just want to talk about the dollars. We're getting amazing return for a little bit of volunteer effort. I don't want to demean the effort of the volunteers. There's 4 of us going out every week and doing these sites. The cost recovery value of the annual sampling project at the 4 sites, the taxonomy, DNA barcode, is around about $360,000. Okay, that's what we're getting basically for free from Paul Hebert and his team. And Paul has recently said he's got enough money also to do another couple of national parks as well. If we can get the take up, we can get the volunteers, so that's a good thing as well.
Would you do more sites in Royal, or would that not be worthwhile?
Well, what we've tried to focus on the habitat types. So there's 5 habitat types, I suppose, isn't there?
Probably a lot more.
More than that.
Or the broader ones like the heath, the wet sclerophyll, the dry sclerophyll, the rainforest.
Yeah.
Okay, so we've done heath, dry sclerophyll, wet sclerophyll. There's two types there, which is the bushy and the grassy. So we've done the wet sclerophyll grassy, but we haven't done the rainforest and we haven't done the wet sclerophyll bushy. Okay, so that might be interesting to do as well. If anybody's interested in getting involved as well, that might be
I'm
nice.
thinking that the major threat in this whole species thing is the spraying of insecticides around. So so would it be more beneficial to find a national park that's well away from agricultural land, say where it's been sprayed?
Oh, that's another research question that could be investigated, of course. But this is very exploratory at this point in time, and so there's all sorts of hypotheses that you could erect. There's more sort of things that we could do subsequent to getting these first results. But I think it's really heartening that the Royal National Park is so biodiverse from what we've done already, and we've only done 1/3 of the samples so far, and we've got all these new species and whatever. So yeah, there's plenty of ways to go, I think, in the near future. So this is some of the international progress within the International Barcode of Life. South Africa, all the national parks are being sampled, and France is about to announce its sampling program soon. Okay, so this is the sort of national commitment that's been made, and that all we've done in Australia so far is 1 national park for half a year. So
you're the only people doing this in Australia?
Yeah, at this point in time, but I'm a promoter for Paul and all his enthusiasm, and so I'll show you what what he's proposing. So South Africa, they're doing it in all their national parks. So far, they've done it in Kruger. They've put in 25 of these nets and they've sampled over an annual cycle, but they're going to do all their other parks as well. And I think it's the National Park Service that's actually doing this on a regular basis. This is the last slide. The vision, barcoding terrestrial invertebrates in every Australian national park. That's what that's what Paul wants to do, and I think that's a great thing to do for Australia, given that other countries, Western countries, are doing the same thing. That we don't want to get left behind. You know, when the technology is available and it's so inexpensive, and it's so important in so many ways, and that's all we've needed. We've needed the heat sealer for the plastic bags that are all provided, and the 95% ethanol, which we get from Macquarie University. So yeah, so that's the talk. And
with the ethanol,
yeah.
How do you go sending it overseas? It's a dangerous good. You have to go through special procedures to get it on an aeroplane to send it to Canada?
You've got to double seal it with some absorbent material inside the second bag. And we reduce the amount in there as much as possible before we send it.
Before we send it.
So
yeah, it's just.
And so we've already sent we've sent quite a few already.
Yeah, I just
with
didn't
no
know
problem.
how many sort of hoops you had to jump through to get it on.
Yeah, it's not that difficult actually. Yeah, not that difficult. So at the end of December, Paul Hibbert's coming back and we're going to give him 2 containers with what we've collected for the rest of the year. And so it's going to be like 100 samples or something.
You'll put it in his luggage and
He'll hand carry it. He'll hand carry it. He's done it before. He'll do it again. He's such an enthusiast. He's amazing. And so,
what is the name of the book that you said we all should read?
Oh,
Planet Insect.
a Planet Insect. Yeah, Planet Insect.
Planet Insects. And
You get it and be enthralled.
And who is?
Do we need to put that on our website?
Steve Nichols.
Steve Nichols.
Yeah, I don't get any money out of this. So just amazed. Yeah,
Steve
can if you like.
Nichols.
Yeah.
And
So-
those images that you showed us before, those beautiful close-ups,
Yeah.
are they your own or they've come from Paul?
Let me think.
Oh.
No, the best ones came from Paul and his team.
Are we able to put them on our website?
If you
or
want
are
to,
they
yeah.
his?
No, he's quite open about this stuff. You can use it if you want to.
Good to
Yeah, do you want to use them all?
label somewhere.
Yeah, I've got quite a few
Oh,
others as well.
T-shirt.
Well, I'll tell him that, it might be a moneymaker.
Yeah.
Make the whole program.
Yeah, so that's where we are at the moment. I regard it as very exciting, and I'm now an insect convert. So I was mainly a fish man, you know, but now I'm really loving insects and realise that I feel a bit guilty for not taking them so seriously beforehand. Because it's such an enormous component of the biodiversity. They're making everything run,
Yeah.
and we're ignoring them and just looking at the avian outcomes or whatever, or the mammal outcomes. So they're all dependent on what these little critters are up to.
Absolutely, not boring at all.
Not bored.
Well said, Dave, well
I fell
said.
asleep. That's great.
Thank you very
Any
much.
more questions for Ross?
Been excellent.
Look, I just want to thank the team, you know. Melissa's here from the team, Sue McTaggart's on the team. Bree McClafferty is supposed to be our liaison officer, but she's a co-enthusiast. And when we're away or doing something else, she gets involved as well. And so it's 4 passionate individuals, very reliable, that are going out and doing collecting. So it wouldn't happen with the highfalutin science that's being done unless we're out doing the weekly sampling in the Royal. Yeah. So I think it's going to put the Royal on the map in another dimension.
Yeah.
Okay.
Thank you.
If you enjoyed this video, please like and subscribe. Consider becoming a volunteer yourself to help keep this iconic gem for future generations.